The first presentation is by Dr. Joan Wilson. She is a senior scientist at the Marshall Institute for Interdisciplinary Research since August 2009. Prior to this, she was with System Biosciences, where she was a PI for a project, Small Business Incentive Research. That project has won an award for developing novel tools for identification and validation of microRNA regulatory targets. Now her group at Marshall, her group focuses on identifying non-coding RNA-based tools for gene regulation and gene manipulation. Without any further ado, here is Dr. Wilson.
Thank you, Venkat. Can you all hear me? Stacy, can you hear me? Thumbs up from Stacy. Okay, a little bit more. Hi, check. All right, I thought today I would start with a quote that I think encapsulates how a classically trained molecular biologist might find herself giving a talk at a cyber infrastructure day. And so besides high-throughput machines such as sequencers, the computer and its associated software has become the instrument to observe the genome. So the purposes of my talk today are really twofold. I hope to get you all very excited about the genome research program that we're establishing here at Marshall. And I also hope that I convey to you how absolutely essential it has been, the cyber infrastructure that Jan has built and the IT team that she has built, and also our interactions with the high-performance computing cluster that Tony has really been instrumental in founding here on campus. Without these computing resources in place, what we have been trying to build at the College of Sciences just wouldn't be possible at all. So my talk today is really divided into 4 parts. I will give a brief introduction to genomics research. I will present to you an overview of the current sequencing technology. I'll talk a little bit about bioinformatics, and then I'll talk some about our current and future projects in our genomics research program. So we'll first start with some definitions. So I know you all are familiar with what a gene is. Sort of conceptually, it is the basic unit of heredity. Operationally, it could be viewed as a series of a certain type of molecule called a nucleic acid. And sort of functionally, it is the basic unit. It encodes proteins or other molecules that carry out functions in cells. But today we're going to be talking really about a genome. So what is a genome? And a genome, it's very simple. A genome is just the full DNA sequence of an organism. And genomics, which is a new field of research, which is really the focus of this talk, is the study of genes and genomes, their structure, function, and regulation. So just to give you a little bit more of a feel for what DNA looks like and the sort of structure of DNA in our cells, I think you're all, oops, sorry about that. I think you're all familiar with the fact that DNA is a double helix, and it is a self-complementary molecule here, and you see the double helical structure here in folding. In a little bit more detail, you can see that DNA is comprised of 4 individual bases: thymine, guanine, adenine, and cytosine. And a really important thing for you to keep in mind for the rest of the talk is that these molecules, or one strand of the DNA double helix, is self-complementary to another strand. So T's always pair with A's, G's always pair with C's. So that is a fundamental feature of DNA that's been important in all the technology we will be talking about. But this DNA does not exist naked inside our cells, or this molecule would sort of go spanning all the way out the building and up Hal Greer. It's actually greatly complexed with cellular proteins that look a little bit like yo-yos here. And the cellular proteins compact this extended DNA molecule down into very tight chromatin structure. And this is probably an image that is familiar to you. This is an example of a eukaryotic chromosome. So for example, the human genome, all of the DNA in a human cell, consists of 23 pairs of these chromosomes. So how big are genomes? The smallest free-living organisms have genomes of about 1 million bases, whereas we humans and all other mammals, our genomes are about 3,000 times bigger. So our genomes are comprised of 3,000 million bases, or 3 billion bases. I know you can't see this, but I don't want you to, so just enjoy the forest while I point out a few of the trees. This is, in one slide, the entire history of genomic research. So it is a new field, as I've mentioned, and whoops, I keep using the wrong button. And everything really before 1990 is essentially the prehistory. And this is sort of the era where I was trained, and back then we called it genetics. So let me just point out a few of the important milestones in the field of genetics. Around the turn of the previous century, the 1900s, Mendel was doing his work, and he was defining the laws of heredity, working with peas. About 45 years later, a team of scientists actually showed us that DNA is the chemical structure of this hereditary material that Mendel had described. And then another 10 years after that, Watson and Crick solved the structure of DNA and showed us that it was a double helical structure, which solved enormous numbers of conceptual problems in the field, and also enabled the development of the first type of DNA sequencing technology in 1977. So this is sort of old school. It's called the Sanger method or directed sequencing method. But in 1977, it became possible for us to sequence a piece of DNA one base at a time and determine the actual chemical structure. This rapidly enabled, in 1983, very important milestone was the first identification of a human gene that was associated with disease. And coincidentally for everyone in this room, that turned out to be Huntington's disease. Very rapidly after that, in 1986, the second gene associated with human disease was identified. This was muscular dystrophy, and 3 years later, the gene for cystic fibrosis was identified. And so, in the years, late 1980s, the federal government started getting very excited about, like, okay, well, we've sent a man to the moon. I think maybe we better sequence the entire human genome. So this was an initiative. The Human Genome Project was an initiative initially formulated by the Department of Energy, and it was launched in the late 1980s. And then obviously the objective of the Human Genome Project was to sequence the entire human genome, and they had devoted $3 billion to this effort. So what is expanded here in this next row is what happened in the first decade of genomics research. So this is 1990 out to 2003. And I'm only going to hit a few highlights here as well. In 1995, the sequence of the first the entire genomic sequence of a free-living organism was sequenced. This also happens to be the free-living organism with the smallest genome that we've ever found. In 1996, the yeast genome was completed. The next year, E. coli was completed. The next year, the worm, a very popular genetic model organism, it was sequenced. 1999, human chromosome 22 was fully sequenced. So it was the first human chromosome that was completed. In 2000, we got the complete genome sequences for 2 more model organisms, the fruit fly Drosophila, and also a very popular plant genetic model, Arabidopsis. And then in 2001, about 10 years after the inception of the project, the draft sequence of the human genome was reported. So I'm sorry Mr. Wellman has left, because this is a classic example of what he described as a horse race which was won by technology, with a twist. So the draft sequence of the complete draft sequence of the human genome was published simultaneously within a week in two of the largest journals in the world, and these were the results of 2 separate groups' efforts. So one group was led by Francis Collins at the NIH. So this was the project that I've mentioned before, the Human Genome Project. It was started by the Department of Energy, and over the 13 years it took to complete the project, the institute was now, the institute that was funding the initiative now had become part of the NIH, and it's the National Human Genome Research Institute. So this effort cost $3 billion over 13 years, and it used the sort of classic, essentially used the classic method of sequencing that had been developed in 1977, and in a massively parallel fashion. This character on the left is a very interesting guy named Craig Ventnor. He's a sort of serial bio-entrepreneur. He was working at the NIH when the government's initiative began, and he became very frustrated at the pace of progress of the research. So he went off and founded his own company, Celera Genomics. They invented this brand new technology called the shotgun method for sequencing, and essentially they were able to complete the human genome sequence at the same time and for about 1/10 what it cost the government to do. So this was the horse race, and there's no question it was won by technology. Okay. So all of this is going to bring us up to present. This is now 2000 to sort of current. And all these advances in technology and computing that enabled us to complete the sequencing of the human genome have also impacted the other animal kingdoms. So this is the painting by Jan the Elder, Jan Bruegel, in 1613, and it shows the entry of animals into Noah's Ark. And the reason I show it here is to illustrate the fact that we've gone in essentially the last 10 years from having the complete genome sequences of 38 bacteria and only 4 eukaryotes, all of which were popular model organisms, to having the complete genome sequences of more than 4,000 bacteria and more than 250 eukaryotes, including horse, tiger, sheep, cow, lion, human, a number of birds, fish, and reptiles. So it's been in incredible progress. And as John was alluding to, the cost of sequencing a genome is now going down, has gone down quite a bit. So for example, 10 years ago, it would cost you $300 million to do it. Today, we can sequence your genome for $10,000. I better hurry up, huh? Okay, so here's the future, and I show a picture of this man. This is Eric Lander at the Broad Institute, and in 2007, he was saying within 15 years, every patient in the clinic will have a complete genomic workup for a couple of $100 per patient. So, you know, in today's money, this is in 10 years, and so this impacts everybody here in the audience. In 10 years, probably your entire genome will be sequenced by your doctor. And then I don't really have time to go through it, but here are some of the newer initiatives, and you know, just to sort of mention one in particular. One genome, you know, wasn't enough, and now we're after 1,000 genomes because we really want to provide a comprehensive, get a comprehensive understanding of the whole spectrum of human genetic variation. Okay, so I'm going to whip through the sequencing technology, hopefully fairly quickly. I want to show you. Again, first, just because it's a general audience, a couple of definitions. What is next-generation DNA sequencing or high-throughput DNA sequencing? It's really just a fast, high-throughput method to determine the order of bases of DNA. And almost everyone today uses this newer shotgun method. And rather than doing it in a directed way in which you sort of take your genome and you put it into parts and you know where this part comes from and you sequence it first and then you do the next part. What you do here is you take your entire genome and you just sort of shear it into little bits and you mix it all in together and then you sequence it, you sequence individual bits randomly. I was going to give you a little analogy to War and Peace, but I don't have time for it. But I think you can see if you start thinking about that. Okay, even sequencing the genome in bits where you know where it comes from and then you get the sequence and you assemble it all back in the end, you can imagine that requires a fair bit of computational resources. Doing it this other way where you sort of just make a big pot of DNA and you sequence everything randomly, I think you can start to appreciate all of the computing requirements that are going to be required to sort of get back to the complete genome in the proper order. So, so I'm going to try and quickly run through this. And so the sequencing is really accomplished in 4 steps. you prepare these big libraries of short fragmented DNA pieces, you amplify them so you can see them, and then you actually do the sequencing reaction. And I apologize, I'm sort of looking at my watch. I'm going to have to run through this part really, really fast. So if you don't get it, you can come talk to us later or come to our lab. We can show you how it all works. These slides were all prepared by the company Illumina, whose machine we purchased and whose technology we use. And so even library preparation occurs really in these 5 sequential steps. The first thing you do is take all of your DNA and you chop it up into little pieces. You have to fix the ends and add these little A overhangs so that onto these random fragments of DNA you can ligate these common sequences here, which you'll see why those are important. We then go through a sort of physical separation process. This is supposed to represent a gel where we separate the shorter pieces, run down these lanes faster, and they'll be around here. The larger pieces that have the adapters ligated are going to run at a slower position, and we cut these out so that we're sure that we only have pieces of DNA that have all of the bits on them that we need to run the next set of chemistry. So the second phase is to amplify each of these individual fragments in the library in a spatially restricted way. And you'll see why that's important. And this is all done on a machine, which I was showing you there. It's called the CBOT. So again, this sort of process can be broken down into 4 steps as well. And it is all accomplished on a flow cell that goes into that instrument. And your sample gets loaded into each one of these 8 lanes, and it flows across the cell. And what you're doing is capturing bits of your DNA fragments. You're creating lawns of DNA fragments in each one of these lanes, which have some amount of physical separation so that you can then perform the sequencing reaction. So we'll go through this really, actually, yeah, I'm going to have to whip through this. We'll go through this really quickly. But you flow your library fragments across the cell, and they stick to complementary sequences that are immobilized on the flow cell. You then make a copy, and you get rid of that strand, and now you have a copy that is covalently bound to the cell. The way that amplification of these individual molecules is accomplished is through a process called bridge amplification, and I'm just sort of letting it run. It's an iterative process that I don't have time to go into too much detail, but essentially what you want to accomplish once you have individual DNA pieces spatially isolated in a lawn across the cell is you want to make about, per every initial one you captured, you need to have about 1,000 of them, 1,000 copies in a small spatially restricted place. And once you've accomplished that, you block these sequences so they can no longer be extended, and you hybridize on what will be the primer that you're going to use for DNA sequencing. Okay, so when we do DNA sequencing, we're going to focus in on this region, and we will actually be adding nucleotide bases here to the complementary strand here, and we will identify them, and that is how we're going to actually get the sequence of this strand here. So let me just move. All of this is accomplished now in yet another instrument, and that is, this is the DNA sequencer. The flow cell goes in here, and everything inside the box is really just sophisticated fluidics and lasers to do the detection. So let me sort of try to go through how that works. So now we're focusing in on our little primer and the DNA that we want to sequence. And across the flow cell, you float all 4 bases. The proper one is going to stick, and then you excite it with a laser. And in this case, it glows green, and that tells you you've incorporated a T, which means that this base is an A. And so again, this is an iterative. So then you unblock it, you bring in the next one, unblock it, bring in the next one, excite the laser, and you get the sequence of your DNA. Okay, so that's just an overview. That's how the technology works. So now, I guess I wanted to say just one more thing about this, that while our instrument is running, so in one run, we can sequence an entire human genome. The data from one run is about a terabyte. And so the way we've set up our instrument over there in BBSC is that in real time, our instrument, through Jan Fox's Internet 2 connection, it sends data over to the IT department here in Drinko, and Ed and his team have set up our server, which deals with all of our data storage and all of our data transfer. And without them, we wouldn't be able to do the sequencing runs that we do. So the next phase is, now that we have 100 million reads, 200 bases each, or 50 billion bases worth of information in short fragments, how do we get back to a genome from that? And this is really the art and science of bioinformatics. So just some definitions here. Bioinformatics is the science of managing and analyzing data using advanced computing techniques. And obviously, it's especially important in analyzing genomic research. Another view of it is the merger of biotechnology and information technology with the goal of, revealing new insights and principles in biology. So it's managing the data and getting valuable information from the data. Okay, I'm going to skip these. So I'm just going to give you examples of two of the sort of bioinformatics challenges that we face. This is actually the more simple case. So for example, if you have a reference genome, so we know the human genome's been sequenced. So say you're sequencing a patient who is presented with a certain disease, and you want to understand if this patient has a mutation in a disease that's associated with that. Mutation in a gene that's been associated with that disease. You can sequence the patient's genome, which you see here is visualized, as I've alluded to, as these sets of overlapping reads with short bits of sequence. And you can align them to the reference genome sequence, which in this case is the human genome. And you can see, for example, this person has mutations in these two places here in what would represent a gene. And this may, for example, predispose this patient to a certain disease, or it may actually recommend a useful course of therapy for that person. So that's sort of best case scenario when you're just mapping to a reference genome that's already been sequenced. A lot of the projects we do involve organisms whose genome sequences aren't available at all. So we have to do what's called de novo assembly. And this is where additional computational challenges come in. The way we meet these computational challenges is I don't even have a computer in my lab. So Jan was sort of alluding to this too. I don't have a computer in my lab that has the processing power to even host the software, the third-party software that's required to do this sort of computing. So this is where Jack Smith and Tony and the high-performance computing cluster comes in, because I am able to putty on into their machines where they host the software that runs these analyses. And I can set up an analysis, go out and drink a cup of coffee, 20 or 30 minutes later, the work is all done on the high-performance computing cluster, and I can go back in and export the results to my own laptop. So this sort of thing couldn't even be done without their help either. So I think, conceptually, de novo assembly, I think it's very simple. You really want to create a contig, which is just short for contiguous, and it is a set of overlapping DNA fragments. And then, if you can take all 50 billion, of your bases that are represented in these short fragments and create these contigs, you will ultimately get the sequence of your entire organism. Okay. So I'm sorry that was a bit of a rush. I'm going to skip that too, because I want to get on to some of the things we're working on here at Marshall, just very quickly. There are a large number of applications ever expanding for next generation sequencing. I don't really have time to talk about some of the ones that we don't do, but in the College of Biology, we're really focused on whole genome sequencing. I'm going to give you one example of each of these three projects, I hope. Okay, so in collaboration with Wendy Trisna, who's in the back of the room, hi Wendy, who is an assistant professor in the College of Biology, we are sequencing the genome of Acanthamoeba. So Acanthamoeba is a common microbe that's present in water and soil, ubiquitously present in water and soil. And one interesting aspect to Acanthamoeba biology is that some strains cause human infections. Here you can see an infection of the eye, and it's not understood why one strain would cause an infection and another strain wouldn't. So our approach is to now sequence the genomes of pathogenic and non-pathogenic strains and see if we can identify genes that are present in the pathogenic organism that are absent from the non-pathogenic strains that are associated with infectivity. So we have teamed up with Dr. Joan E, who's at the Stanford Genome Technology Center, to help us with the bioinformatics of this. Another project that we're working on, this is a Metagenomics project, and this is really being pioneered with Dr. Gary Schultz, also of the College of Biology. And what Gary wants to do is he wants to take, so a metagenome is, it's one layer of abstraction beyond just the genome. It's not just a single organism's genome. It's really looking at the genomes of all of the organisms in an environmental sample all at once in an unbiased way. So just as an example, what Gary will do is he'll take a sample of water from the Ohio River or some other local water source, and we will filter out all the bacteria, all the algae, everything that's living in it, prepare DNA en masse from that organism, and sequence it. So this is sort of metagenomics. And what we want to learn, a couple objectives of this sort of project. One is just to catalog the biodiversity of organisms, in our environment. So we are now in the age, we're no longer Charles Darwin going to the Galapagos and sketching finches, and we're no longer Linnaeus collecting plants. But we are now at the stage where, it's essentially just it's an extension of this, and we are actually now cataloging the world's biodiversity at the level of gene sequence. So, one thing, it's just interesting to know what is the biodiversity there and how does it respond to different environmental conditions. So that's an interest for us here in the state of West Virginia. And then another interest is that if we don't know what's there, we're not, there will be unrevealed there will be metabolic capabilities existing in these organisms that we're not aware of. And until we sequence the genomes and understand what enzymes are present, we may be missing really important biological functions, or we may be missing natural molecules which could be important for bioremediation or that sort of thing. So there are a lot of exciting things that could come out of that project. I don't have time to talk about Dr. Paul Lee's salamanders, but the thing I want to end on is a really exciting project. We started kicking around the idea of what would be an important genome to sequence here at Marshall. And of course, Marco is our mascot and we are the herd, but do you know what we are the herd of? Well, we are a herd of American buffalo. So there's some very, so we went and contacted Jim Durr, who is a professor at the College of Veterinary Science at Texas A&M, and he is Mr. American Buffalo. So he's a conservation biologist. He's really interested in preserving the integrity of the few remaining natural wild bison herds that exist in this country and Canada. And so integration of cow genes is a real problem in the wild. And so we said, Jim, why don't we sequence the genome of the bison? And he said, well, it's funny that you come to us at this time because I'm just putting together a consortium with the USDA and the Canadian Genome Foundation to do exactly this. So the informatics on this project are going to be done at Cornell University. Jim wants to, so this is Templeton. This is going to be the type specimen for the bison genome. This sequencing is going to be done at the University of Iowa. But Jim wants to sequence an animal from a different herd at Yellowstone, possibly 2 Canadian animals, and he has a hide from the 1850s he'd also like to sequence. So we don't know which one we're going to get, but we at Marshall are going to sequence one of those genomes. So we thought that would be an exciting project to sort of build an undergraduate course out of and start getting some students' hands dirty in actually dealing with analysis of genome sequences. And I just wanted to put this slide up, and you can't really see it very well, but does anyone in the audience recognize this fellow? Okay, so we're really excited by the possibility of maybe some private sponsorship for this project. This is Ted Turner, and he has a buffalo herd himself, and he wants his animal sequenced too. So we're working very hard with Jim to sequence Mr. Turner's buffalo. So we're having a lot of fun with the project, and I've alluded to this before, but I want to make it more explicit. We could not do anything at all if we did not have the resources of the IT department here. Our key people in the IT department who have been extraordinarily helpful to us have been Ed Eric Tingy and Gerald Hesver. We also couldn't, and this is just managing and storing our data and accessing it and sending it out to our bioinformaticists. We here on our own couldn't analyze any of the data at all if we didn't have the help of Jack and Justin at the High Performance Computing Cluster that hosted all the software that we do to do it, because we just don't even have computers that could manage it. Cassie is the one who, I say we, I've met Cassie all along. She does all the work. Elmer is the chair of biology and the supervisor of the program. And I'm actually on volunteer from MIR. And this is not my day job, this is my avocation, but I am devoted to it because I think it's really important. And I have to thank my boss, Eric, who is the director of MIR, who allows me to devote time and effort to this project. So thank you, and I'd be happy to try and answer your questions.